Stem cell donation begins with selecting healthy donors, followed by a non-invasive extraction process. In Mexico, rigorous quality checks ensure the best stem cells for successful transplantation and recovery. Let's get into the details of the process with Dr. Anand Srivastava. He received an Outstanding Achievement Award from the Society of American Asian Scientists in Cancer Research. He was also awarded with the USA Congressional Award for his contribution in the field of stem cell science.
In general donor selection, there is a specific guide for the donor selection that is established by all the countries, including US or India or any country. So you have to go through a specific test, like whether that person has any kind of viral infection or not, sexually transmitted diseases or not, any kind of other disease. So there are a list of the diseases. So you have to go through all these things and then select the donor. Of course, you have to get consent of the donor that if he wants to give or not. And then umbilical cord, yes, same way. You can take the umbilical cord from after the baby born. You generally throw the cord. So you can take the cord, but of course you have to screen for the diseases that this cord is free for any kind of genetic or any kind of disease which may be transmitted. So that's the way to select the donor. In general, we use adipose-derived mesenchymal stem cells. And this adipose means you can take maybe 50 to 100 milliliter fat cell, and that is not the kind of liposuction, it's lipoaspiration, we say. Just mini lipo, we say. So you don't need to go for general anesthesia or all this, just using the local anesthesia, and a plastic surgeon can take out 50 to 100 milliliter cells. It goes in the lab, and we have a state-of-the-art lab every single place. This fat cells goes in the lab. We have scientists in the lab, and he follows the protocol, how to isolate the stem cells, and then it goes for the quality check and all these things. Each and every single cells goes through the quality check, and then it goes back to the patient. The quality check is several steps actually. The most important thing that how many cells are live. So when you are isolating the cells, and most of the people have this problem. They don't know how to isolate because when cells are out of the body, there is no immune system. Cells are totally open in the atmosphere. So there is specific guidelines, specific instruments where you have to isolate the cells, and because there is no immune system to protect the cells. Then how many cells are live? That you have to check. Then because you are trying to get a specific kind of stem cells, then there is specific markers to check each and every single cells that these cells are really stem cell or some other cells. So there are several stringent quality related steps which anybody has to go through and which we have well established in our labs.
MTC is with you every step of the way. For your medical or dental trip, contact MTC today.